are good to get started. I apologize for being just a little bit late getting going today. I adopted another bird about a week ago, and today was sort of the first day they're all together. And needless to say, it did not go well. So I needed to do a little bit of emergency first aid on myself and then separate them. So hopefully they're better when I get back. All right, so for today, we're going to do a little bit of review of what we talked about previously in how neurons fire, how they produce action potentials. And then we're going to move on into talking about glial cells. And then once we're sure we have those basics, we're going to add some extra stuff to our understanding of how neurons communicate. And I find that this starts going into maybe a little bit more detail than you've probably seen in other classes, unless you've done cell bio or a neuro course. So we start adding sort of small things in here and there. So we had started off with this is generally what we think of when we look at a neuron. We did specify that we are going to mention there being other kinds of neurons. So this one here is a bipolar neuron. We've specified that it's a sensory neuron for us. And it's bipolar because there are two offshoots from that cell body or from the soma. So we have our dendrites as a single branch off that cell body, and then the axon as a single branch, which is as expected. But when we looked at sort of our other options, we can have a unipolar neuron, so only one projection off of the cell body, and that connects to both the dendrites and the axon. So uni meaning one, therefore one projection. We covered the twos. We can have a multipolar motor neuron where there's lots of dendrites connecting into that cell body, multi just meaning multiple. And then our interneurons are those weird ones where they mostly just make connections and they don't follow the same rules that we typically see in our sensory and motor neurons, and we kind of left it at that. So the part that I wanted to go back over again was, here, where due to my own mix-up, we had talked about this in the wrong direction at first, so I wanted to go back over that with the correct arrows again, just to make sure we're all on the same page. So we talked about that potential across the membrane. The fact that there's a difference in electrical charges on the inside and the outside of our, I'm drawing a blank there, phospholipid bilayer of a membrane. There we go. So we have sort of differing charges, differing molecules that are stored inside and outside, and that leads to an overall net voltage across the cell membrane. That just means that in this case there are more, or it's relatively or comparatively more negative inside and comparatively more positive outside. And what establishes this is actually the sodium-potassium pump. So this is an active process. It's driven by using up ATP. And so what it does is that these blue sort of bean-looking things in the middle, they have two types of kind of like holders, clamps, and they can hold on to either potassium or sodium molecules. And they hold on to and release them in a complicated manner that we just summarize as they pull in two potassium ions and they push out, which is, I apparently didn't fix the animations, they push out the 3 sodium ions. So we end up with more of those red sodium ions outside. That's where we're getting more positives outside. And then because we're only bringing 2 of our potassiums, the K pluses, coming back inside, it's moving positives in both directions, but more positives are going out than coming in, and that's how we end up comparatively negative on the inside. So what we're doing is looking at the difference across that membrane, and that's why the movement of positive ions is actually leading to a negative voltage across the cell membrane. So that's the part that I wanted to go back over, because that sets everything up where we have lots of our sodium outside and lots of our potassium inside. And because of those forces that we were talking about, where they want to diffuse to an area where there are lower concentrations of the same kinds of ions. So the sodium wants to get away from the sodium, the potassium wants to get away from the potassium via diffusion. And we also have that issue with charges, where the positive charges want to get towards the negative and the negative towards the positive. There's a lot of tension, there's a lot of potential energy stored across this membrane. And so that is our setup, where once that has been established, we can look at how an action potential is triggered by releasing that stored energy. And this is where our ion channels come into play. And so there are different kinds of ion channels. When we refer to them, we usually call them by the name of the ion that they're designed to work with. So we can have sodium ion channels that will let sodium pass across the cell membrane in the direction it wants to go. And because sodium is outside and wants to be inside, when a sodium ion channel opens, the sodium will come inside. And then we can look at how the charge across the membrane changes with that influx of lots of positive charges. And we specified we can have chemically gated or electrically gated ion channels. So they can be triggered by a change in the potential across the membrane. If there's a change in sort of electrical potential energy, they can be triggered to open. But there are some that can also be triggered to open by the presence of other chemicals. So whether it's individual molecules or atoms or whatever. So we started mostly looking at the electrically gated ion channels. And then the big thing that we had sort of started adding to our conceptualization of how an action potential happens is the idea of a local potential. Because usually in most classes, they just talk about action potentials and they kind of skip what leads up to that action potential occurring. So we're starting to dabble into some of those more complicated concepts. And we're talking about local potentials, which are partial depolarizations that can then trigger an action potential. So these local potentials are graded potentials. So unlike our action potentials, which are that all or none, once they fire, they always fire at the same sort of amplitude and they look exactly the same, these local potentials are graded, meaning that they'll vary in magnitude based on what's stimulating them. So more incoming excitatory signals means a larger shift towards the positive. More incoming inhibitory signals will mean a greater shift towards the negative. And we're looking for those local potentials to build up to the point that they might trigger an action potential. So if they can hit that threshold, they can trigger an action potential. All right. And so this was our diagram where we showed what was going on when sort of the cell is at its resting state. We have our standard average minus 70 electrical potential at rest. And we mentioned that resting potentials, though we usually see it as minus 70, they can vary based on what kind of cell you're looking at and where it is. So we just use that general rule of thumb. And here, our threshold is minus 60, and that's because, again, our standard average threshold is going to be 10 more positive than your resting potential, more or less. And so once we cross that threshold, we trigger that action potential. It's a sufficient difference in charge to trigger those ion channels to allow the sodium to flood back into the neuron, which is why we see that massive spike of the charge there, the electrical potential changing up to positive 30 here. And that's because lots and lots of sodium has flowed in. And because of that momentum of everything flowing in, we end up being more positive than neutral. It's like it's gone too far because they're all flooding in at once. And then once that has happened, once those are in and we've overshot to positive 30 or 40 or whatever that cell does, then that change gets corrected by the potassium ion channels opening and allowing potassium to flow back out. So At first, this is when we started introducing our next concept, which is not an absolute refractory period, but a relative refractory period. It's on whether the potassium channels and the sodium channels are still open or if they've closed. So, actually, yeah, I did have a slide for that. So we usually talk about the absolute refractory period where we now know that the sodium ion channels are still open, meaning that sodium can still come into the cell freely, which makes it impossible to trigger a new action potential. Because they're already open, all of the sodium's already inside, there isn't enough sodium outside to trigger a new action potential. So an absolute refractory period is a hard, it cannot happen. Whereas once we hit this relative refractory period, so after a couple of milliseconds, sort of 1 or 2 milliseconds, we then hit into this relative refractory period, where the sodium ion channels have closed and could potentially reopen to allow another influx of those sodium ions, but the potassium channels are still open, which means that there's still a movement of those positive potassium ions out. So we have to have a large signal in order to trigger those sodium channels to open again, because we haven't really reached our equilibrium yet. So during that relative refractory period, we say that it could happen if the signal was strong enough, but we need a very strong stimulation to overcome that in order to produce a new action potential. And so this was the first of our ways of influencing our rate of responding. So the absolute refractory period is the hard cutoff of how soon we can have another action potential. And then the relative refractory period gives us sort of a span in which more intense stimuli can produce another action potential. So if you're getting action potentials closer together, then the distance in time between that absolute and relative refractory period, then you know that you're getting a stronger than normal signal. So if we see closer together action potentials, if we know that those action potentials are happening within that relative refractory period, then we know that it's a stronger signal and our brain can interpret that as necessary. And so that led us to that idea of the rate law, where the axon encodes stimulus intensity not in the size or magnitude of the action potential, but in the rate of action potential, so how quickly they're firing. So more action potentials closer together means a more intense stimulus because of those setups with our refractory periods. Okay, so that's all review there. And like I said, with topics that are sort of adding these extra details, I like to go back over them at least once just to make sure that we're on the same page, because hearing it multiple times tends to help it sink in. I've also posted a couple of videos that I find just helpful in order to visualize things like how an action potential propagates down a neuron's axon. I find, for whatever reason, it didn't quite sink in for me, because you always see these static images, but then having them show you like a simplified version of an axon and how the charge propagates along, that was really, really helpful. So I've shared that on our Moodle. And then I have another one that just gives a crash course in neurons, because sometimes hearing it from a different perspective is useful. So those are there, and if I find any other videos, I'll let you guys know about them. But that gets us to the point where we can start adding on to our understanding. So we're going to talk a little bit about glial cells, and then we're going to start getting a little more in depth about how this communication can be interpreted and how it gets more complicated than just firing or not firing. But let's start off with glial cells. And these are non-neural cells that provide a whole bunch of different supporting functions to neurons. So in most psych classes, when we talk about glial cells, we just say that they're the helper cells around neurons, and we leave it at that. I've gone as far as having a slide that looks kind of like this to talk about glial cells and then never coming back to them again. But for this class, if we're talking about brain and behavior, they're obviously going to be really important. We want to get a more in-depth understanding of what's going on, so we're going to talk more about them. And I found it kind of interesting to know like the roots of some of these words. If you've taken enough bio courses, you've learned that the Greek and Latin roots of words really helps you remember what they mean. So for glial cells, the root or the word is glia, and it's from the Greek word for glue. So our historical interpretation of glial cells is that they're the glue holding and supporting our neurons together. As we'll find out throughout this chapter, it's a little bit more complicated than that. They're not just sort of support structures in like sort of a structural sense, they're also helper structures, and they also do functions themselves. So we'll talk about a bunch of different roles that these cells take. But before we talk about different types of glial cells, I do also want to talk about myelination and conduction speed along an axon, because glial cells are very tightly tied in with those concepts. So you might already know about myelin and our myelin sheath. So myelin is a fatty tissue that wraps around the axon of a neuron in order to insulate it. So I always interpreted this as insulating, kind of like you insulate a wire. So if you're thinking of electrical wires, that plastic tubing around it that keeps the electricity from leaking out, that's what the myelin sheath is, the end. But it's, again, a little bit more complicated than our simple model might imply. So this keeps the cell separate not only from the extracellular fluid, the stuff that surrounds the neuron, but it also protects it from any spillover, any potential signals coming from neighboring neurons. So this keeps everything separate and isolated and kind of allows that signal to stay where it's supposed to. We'll even see that some of our glial cells help keep our neurotransmitters in the synapse because the method of communication is just sort of to dump chemicals out into the world, and they could go anywhere, so the glial cells at a bunch of different levels help keep the signal where it's meant to be. And so if you know about myelin and the myelin sheath, you probably also know about our nodes of Ranvier, and they are the small gaps in the myelin sheath. So they're only like a fraction of a millimeter wide, but they're little breaks in that insulation that allows the surface of the axon, that cell membrane, to interact with the environment as it would normally. And so the presence of these nodes of Ranvier actually leads to something that they call saltatory conduction. And I remember this one because the word saltation means that something appears as if from nowhere. So saltatory conduction is a form of transmission where our action potentials kind of jump from one node to the next. On the surface, it looks like nothing's happening in between. And this is actually because that myelin is wrapping around the surface of the cell, and it's actually preventing any kind of action potential happening underneath that fatty tissue. In fact, underneath that, there aren't, say, ion channels that can allow cells to flow in, which can be problematic for degenerative diseases like MS, which causes the degradation of our myelin sheath. So as that myelin is eaten away, not only do we have issues with conductivity because now stuff's exposed to the cell fluid, but also there weren't ion channels underneath the myelin, and so it's impossible for them to start conducting again, and that is one of the major factors that leads to that loss of signal with MS. Actually, one more thing I wanted to mention before I moved off of this slide. With conduction speed, myelin is sort of it's like the engineering equivalent of allowing these things to go more quickly. There are other ways that you can speed up conduction. If you look at, say, giant squids, so they are huge, And a lot of people who study neurons like to study giant squid neurons because their axons are massive. I think they're as large as like a 0.5 millimeter, which is huge when it comes to neurons. And the larger the diameter, the actually faster it allows that signal to propagate, something to do with it allowing more ions in at that location and then being able to push along faster. So if you wanted a very, very fast signal, you could just make your axons larger and larger, but that becomes problematic because we remember how many neurons are in our brains, and if they were all significantly larger, that would cause lots of problems. So we're kind of stuck with the diameter that we have now. We will see a push with evolution towards larger neurons to a point if fast responses are important, which, if it helps you react quickly to things like predators and catching food and all of that, it's usually pretty important. But we do have those limitations. And so this insulation, this myelin thing, is our alternative solution. And it's actually the equivalent of having our diameters much, much, much larger, even like 100 times larger than what they could have been. That's the equivalent that we get just by having these myelin sheaths, which is pretty cool. So we found an alternate way to speed up conductivity without having to have giant neurons. All right. And so I've already kind of touched on these, but I wanted to summarize the benefits of the myelin sheath all in one place. So the first of these is the idea that it reduces capacitance. And capacitance is a fancy term for some electrical effect of the membrane that actually just slows down the movement of ions. So if we didn't have this myelin sheath, then we would have to have that action potential propagated at each point along the axon's length. But by covering most of that space, by not allowing the action potential to propagate at each individual point, we can sort of hop ahead. We can use graded potentials to get between the nodes, and it is sufficient to get to the next node and trigger a new action potential, which then sort of carries along enough of a charge to trigger the next one. So by skipping that, we're reducing capacitance, we're reducing that slowdown, and therefore increasing the speed of conduction. Also, by having our all or nothing action potentials at the nodes of Ranvier, that gets us to not have a reduction in signal. So if we were just using graded potentials, as time goes on, you might lose some signal because you're trying to travel ions and changes in energy across distances. So as time goes on, you could find that the amount of signal reduces. But the fact that our action potentials occur always at the same magnitude with the same intensity as long as you've crossed that threshold means that we continually regenerate the strength of our signal. So no matter how many times it has to fire again, you're not going to lose out on any of that signal. So we can skip ahead, so we're firing faster. There's no loss of signal because they're spaced out in such a way that they will always trigger the next node to fire as well. And the last of our benefits is that because we don't have these ion channels and pumps and everything that's triggering and maintaining these action potentials under the myelin sheath, those areas get to save a lot of energy. Because as we mentioned, using pumps to reestablish a balance of ions across the membrane uses up a lot of power. And so if you don't have those, if you don't have to maintain that resting potential, you get to save that energy. So there's lots of benefits here to having a myelin sheath. So it's not just the fact that it allows us to have faster transmission, but that is one of the biggest benefits. All right, so now that we've talked about myelin, we can actually go back and discuss why myelin connects to glial cells. So there are two types of glial cells that actually produce myelin, and they tend to be divided up by type based on where they're located. So we have oligodendrocytes, which are glial cells that produce myelin, and they're in the brain and spinal cord. So that's our central nervous system, which we'll get into the breakdown of the nervous system in a later chapter. So about 70% of the glial cells that are in our brain are these oligodendrocytes. So having a lot of myelin in our brain seems to be very, very important. We also have Schwann cells, which are glial cells that produce myelin in the rest of the nervous system. So it's nice and easy to tell the difference between these two because of the location. So if I comment that there is a myelin-producing glial cell and it's in the brain, you can assume that it's an oligodendrocyte. If I state that it's in the arm, then you know that it's a Schwann cell. And just for a visual of what these oligodendrocytes or Schwann cells would look like, we have two here. So the one in the middle is labeled as an oligodendrocyte, and it connects to different axons. And then we also have the Schwann cells, which wrap around axons outside of that central nervous system. Just a visual for you. As I said, these aren't the only kinds of glia. They're just the ones that are involved with the myelin production. We have other kinds of glial functions, so there are other glial cells and they do other stuff. So we have radial glia, and these are active during fetal development, and they basically form scaffolds that allow the neurons to migrate into their correct locations. So they're kind of pre-programmed to sit in very specific places so that neurons, as they form, can make the connections that need to be connected in the brain. We also have microglia, which basically give energy to neurons. So they provide energy, they take away cell waste, and they help repair injury whenever they can. So those are more of that support function. And then we also have astrocytes. And this is an interesting one. So astrocytes end up triggering the formation of extra connections with neurons. So they trigger the formation of synapses. So the presence of these astrocytes leads to more neural connectivity. And experimentally, they've shown that if you have neurons that have astrocytes and they've made lots of connections, and you take away those astrocytes, the number of connections between the neurons that remain will actually go down. So they might be maintaining and creating those. Again, not quite well understood, but that's the basic idea. So they seem to play some kind of key role in learning and then remembering what you've learned. But I'm sure the future will show us more about that. All right, so now that we've covered our basics of neurons, our basics of glial cells, now we're going to get into just a little bit more detail about how neurons communicate. So how do we go from action potential or no action potential to something that means something? How do we sort of add to this and get the complexity that's required for our brains to do all the brain things that they do? So you might already know most of these terms, and I think we've mentioned at least most of them, I think. So the synapse is that connection between two neurons. If we call it the synaptic cleft, we're specifically referring to the gap that exists between them. Usually the terms are pretty much interchangeable, unless somebody is being pedantic. We also have our presynaptic and postsynaptic neurons, and that's the presynaptic is the one that is ahead of the synapse. It's ahead of the gap, so it is the one that we are looking at as transmitting. And then postsynaptic is the one that is after the gap, so that is the one that is receiving. So those are good terms to remember because when you're referring to these chains, this transmission of information, we can casually refer to the presynaptic neuron and you understand that that's the one that's sending a signal. And if we talk about the postsynaptic one, that's the one that's receiving that signal. And as a little reminder of what these synapses look like, so the whole thing would be considered the synapse. If we're talking about that synaptic cleft, it's that gap between. So the presynaptic axon terminal doesn't quite touch these postsynaptic dendrites, but it comes very, very close. And so that gap is where there's going to be that transmission of information. And there's also that presynaptic and postsynaptic labeled there, so just a visual for those who prefer those things. All right. And so nowadays, we know that most of the communication that happens between neurons happens by chemical transmission at the synapse. But this is a surprisingly recent discovery. Before the 1900s, or before the early 1900s, there wasn't a lot of knowledge about what was going on. And in fact, they weren't even sure if the synapses were physically touching or not. There was a lot of debate as to whether it was an electrical signal, like what happens within the cells, or is there something else going on? And so the text goes into this neat little anecdote about Otto Louis, who was the one who finally showed that it's chemical transmission that allows signal to transmit between neurons. And the way that he did this was kind of cool. I'll summarize it very briefly, but if you think it's a neat story, the text goes into like paragraphs about it. But basically, he had this idea that if the neurons were communicating in some way that wasn't electrical, if they were communicating with chemicals, then they would be putting those chemicals out into the world. So he took the hearts, the still beating hearts of two frogs, and he bathed them in a saline solution, so something relatively neutral that's going to maintain that charge or the extracellular fluids balances. And with one of the hearts, he stimulated the vagus nerve. And so the vagus nerve is, we have one as well. When you stimulate it, it basically slows things down. So it's a really quick way to calm down, to slow the beating of the heart. Everything sort of decreases. And so his idea was, if I stimulate this nerve and the heart slows its beating, and I collect all of the saline that's bathing that heart, if it's not just an electrical signal, if there's something chemical, it might be in that fluid. So he took the saline from the heart that he had manipulated, and he used it to bathe the other heart, the one that he had never touched the vagus nerve on. And what he found was that the heart rate of the second heart slowed down, just as he had predicted. He also wanted to see if, okay, does it work in the other direction? If I take a first heart and I get it to beat faster, will the second heart also beat faster? And the answer was yes. So through this conceptually simple idea, he showed very cleanly that there is something that's in that extracellular fluid, that's in the saline that's around the neurons, that was triggering either an increase or a decrease of that heart rate. So he could conclude that neurons release at least two different chemicals, and they have opposite effects. One increases, one decreases. So that sounds pretty much in line with what we know nowadays about neurotransmitters and them being either excitatory or inhibitory. And that's where we started from. One thing I did learn from our textbook is that while chemical transmission is still the most common way of transmitting signals, there are special cases where there are electrical signals that transmit between cells, and there are gas transmissions between cells, which I thought was really cool. And both aren't very well understood, but I just thought it was neat to point out that they exist. All right, so for our chemical transmission at the synapse, we're talking about that movement of neurotransmitters from our presynaptic neuron to the postsynaptic neuron. And they're going to do something with that chemically transmitted signals. So we have our vesicles, and these are membrane-enclosed bubbles that exist at the axon terminals, and they're going to store neurotransmitters. So these vesicles are basically like balloons that hold all of the neurotransmitters together. We'll talk about a couple of different ways that we store and release these neurotransmitters in a controlled fashion, but that's in a couple of slides. So the way that these work is when they are triggered, they can migrate a little bit, connect to the cell membrane. Because vesicles are bound by their own membrane, the two membranes can form together and then open up so that the neurotransmitters dump outside into the extracellular fluid at that synaptic cleft. We can have different kinds of receptors. So we have ionotropic receptors. So these are ones that, hold on, let me get my thoughts here for a second. Okay. Let me start by explaining the difference between the two first, and then we'll talk the specifics. Okay, so two different kinds of receptors that are going to work at differing rates. So one of them is sort of a quick, fast but short occurrence, and one of them is more of a slow and longer enduring kind of response. So these receptors join with the ion channels, and they tell the channels to either open. So the first, those ionotropic ones that I started with, they open very quickly and produce an immediate reaction. So they join directly to that ion channel and tell it to open right away. Whereas the metabotrophic, extra syllable, metabotrophic, you can think of it as being metabolizing, where that takes just a little bit longer. There's a couple of extra steps. And here, instead of having a direct and immediate effect on those channels, they're going to open the channels indirectly through a second messenger. So there's usually a second step involved. And because of that second step, it takes a little bit longer for those signals to open up those ion channels. But once they're open, they persist for longer. So instead of having sort of a fast acting but quickly gone signal, we can also have slower acting and longer persisting signals that can exist. So that's one way that we can affect what kind of a signal is transmitted and when it's transmitted. And then when we're starting to look at our excitation and inhibition, we've kind of already discussed these topics, but I want to go over it again with some extra terms and give us a little bit better of an understanding of what's going on a smaller level. So we talked about there being partial depolarization. So that's what happens when you have a slight excitation signal that comes in. So this is depolarization going towards the positive. The charge is increasing. So it's excitatory because it's increasing that charge. And it can facilitate the occurrence of an action potential because it's moving that charge closer to our threshold, which would trigger an action potential. So when we're looking at that partial depolarization, we're saying that we're getting more positive, but it's partial, so it's not sufficient to trigger a complete action potential. We can also talk about hyperpolarization, where we make it more negative, so we can overpolarize. So we go more negative than our resting potential, and by dropping that charge even further, we're setting it up so that it's even harder to trigger an action potential because it's harder, you have to go further to get to that threshold. And it is a good thing to note here that inhibition can communicate just as much information as excitation, even though we typically talk about action potentials as either they happen or they don't happen. It's not quite that simple, which is going to be the running theme for this chapter, and I suppose probably the rest of this course. So we can also have sort of the holding back of signals, and we'll talk a little bit more about this in a slide or two. I can't remember where it comes up relevantly, but a lot of our neurons don't only fire when they receive a signal. They often have sort of a background level of fire, which we've never talked about before and usually gets omitted from classes like this. But again, brain and behavior, we get more in depth. So these neurons, our neurons, tend to fire at sort of a background rate. So a very predictable, slow fashion. And the presence of excitatory neurotransmitters, if we have that excitation, then we see more frequent firing. And then if we see inhibition, we see less frequent firing. So it's not just that it's a yes or a no, it's also a rate. So we can see a significant decrease or a very, very different decrease in rates of firing, and they can mean different things. So it's more than just all or nothing, it's more than just on or off. We're going to start building extra into that too. Okay, so the next thing that we can talk about is what's happening on the postsynaptic side. So releasing neurotransmitters and doing stuff on that side is mostly what we've been talking about, but now we're going to move into how we have stuff happen on the postsynaptic side, after the synapse. So we can look at excitatory postsynaptic potential, or EPSPs, which I will never say that because I'm never going to remember the order. But these excitatory postsynaptic potentials, this is basically when the receptors when they're activated, they open sodium channels and produce a partial depolarization in the dendrites and or cell body. So we've talked about the fact that most of our axon terminals connect at the dendrites or maybe the cell body. That's sort of what we normally assume. So these excitatory postsynaptic potentials, we know it's excitatory, so okay, it's increasing our charge, so we are depolarizing, we're getting less negative, we're going more positive, which means that we are more likely to see an action potential further on. We can also have the opposite, those inhibitory postsynaptic potentials, are IPSPs, and that's when you have receptors that open the potassium channels, so instead of allowing the sodiums in, they only allow the potassiums out. So it's kind of like it's forcing that, make things extra negative. And as the name implies, we're forcing it to be inhibitory, we're producing hyperpolarization, we're making it more negative. And so these are the two things that can occur. They can increase or decrease the charge by acting on those two types of ion channels that we've already been talking about. So excitation is kind of like the start of an action potential where you let in some of those sodium ions. And inhibition is kind of like the end of the action potential when you're trying to shut it all down and you're letting out some of the potassiums. So that's what's going on there. And so if we have these increases and decreases, they must interact in some way. And so we can call this process postsynaptic integration. And it's because you're combining these different excitatory and inhibitory signals. So we have two different kinds of summation, two different ways that we can add these signals together. The first of these is spatial summation, which combines potentials that are occurring at the same time at different locations on the dendrites and the cell body. So we can have lots of different axon terminals from lots of different neurons connecting to our postsynaptic neuron. They can be connecting at different places, but it's all one cell. So we can change the concentration of ions inside at multiple places, and it has an overall effect on what's going on across the membrane of the whole cell. So we can actually hop to our next slide here, where we look at what happens with different kinds of excitatory and inhibitory signals. So if they go up, they are the excitatory ones. So 1, 2, and 3 are all involving excitatory signals. So number 3, let me just double check what all the numbers are here. Because number 3, which is a spatial summation of our excitatory signals. And we have, at that point, it looks like 3 little bumps that are all together. So because those signals are coming from different places, there's a little bit of a delay as to when they arrive at the place that we will start firing. But they will all get there around the same time, and because it's being driven by signals from different places, that's going to then add together, and in that case, it's crossed the threshold. Those 3 different signals was sufficient to push us across the threshold and trigger an action potential. We can also have temporal summation. So instead of just looking at different places that are triggered, we can combine potentials that arrive sort of in the same place but at different times. Basically, there's a little bit of a lag in those ions resorting themselves. So once a signal has occurred, we're going to persist in a either slightly higher or slightly lower charge just for a little while. And if these neurons are happening fast enough together, we can actually have two happen back to back that would add together. You can have more than that. And we can look at this again. Oh, the numbers are for after. That's why I was confused. Okay. So it's showing that 4 was the one I was talking about. I thought they were at the beginning instead of at the end. It's the same basic idea, where it's just because they're at the same time, they're kind of squished closer together. But with the same location, they're a little bit more distinct in terms of bumps. I'm not going to show you a graph and ask you to interpret it. I was hoping this would just make it clearer, but again, that kind of backfired. All right, and so that can go in the positive or the negatives. So the spatial one is focusing on different places, same time, and the temporal one is focusing on same place, different times. And I just want to double check if there was anything else I wanted to say here. Yes, okay. All right, so next we can start looking at things like, what do you do after you've transmitted that signal? Once we've increased or decreased whatever we needed to increase or decrease across that gap, now what? And you might remember that there are a couple of different ways to remove those neurotransmitters. One of the most common ones is simply reuptake, where those transmitters get taken back into the terminals that they were released from and put back into vesicles for future reuse. So there are specialized transporter proteins that will go out and pick up those used neurotransmitters and bring them back to the presynaptic neuron terminals and allow them to be put back into vesicles and reused. That's a fairly efficient process because you don't have to make new neurotransmitters. At some synapses, they have glial cells that are nearby, astrocytes, which can pick those up, kind of like extra debris that they're cleaning up and recycling, so they can turn it back into their components and use it for other stuff later. In some cases, you'll see them inactivated by enzymes. One of the most common ways for this to work is for an enzyme to be designed to break down a specific neurotransmitter. So it'll usually cut that neurotransmitter into pieces that no longer function as a whole. And then, of course, it can be recycled and reused potentially. All right. So we focused there on our postsynaptic neuron, and now we want to bounce back and talk about the presynaptic neuron again, this time talking about whether we're increasing or decreasing the release of neurotransmitters. So we can talk about a presynaptic excitation, and so again, these words should start making sense because we're using them pretty consistently, just in different combinations. So we're talking about the presynaptic neuron, so we know we're looking at the one that's releasing the signal across that synapse. We're looking at excitation, which is an increase. So we're going to increase that presynaptic neuron's release of a neurotransmitter. So if you trigger presynaptic excitation, more neurotransmitter is released. We can also have presynaptic inhibition, which decreases the amount of neurotransmitter that's released. And of course, you want to keep in mind that different neurotransmitters themselves can be excitatory or inhibitory. So we're starting to see that there's lots of moving parts, lots of possible combinations here. So we can have presynaptic excitation, so more release of a neurotransmitter, and if the neurotransmitter that's being released in larger amounts is inhibitory, we can actually see a decrease in activity in the postsynaptic neuron. And it actually does make logical sense that you can have excitation leading to inhibition because we're evaluating the excitatory or inhibitory properties of sort of each step separately. So when thinking about stuff like this, I find I don't do a great job at keeping all of the directions straight. So what I would do is I would draw a little diagram, and I'd have sort of a neuron, a gap, and then a neuron, and I would draw little up and down arrows for what each stage is doing and try and figure out what that means. I don't know if that works for you guys, for how your brain thinks it through, but for me, that's typically how I approach things like this. So I would think about, okay, we have our presynaptic neuron, it's being excited, so it's increasing, it's producing lots of neurotransmitter. What is that specific neurotransmitter going to do? Okay, it's an inhibitory neurotransmitter, so it is going to have an inhibitory effect on our postsynaptic neuron, which is then going to increase our polarity. We're going to go to hyperpolarization because we're getting more negative, and that's the way I would think through it. All right. And then we can also have something called autoreceptors. And this is, again, just another level of complexity. And these are receptors on those presynaptic terminals, which will pick up and detect the amount of neurotransmitter in the synaptic cleft. So these are basically like little survey cameras that tell the presynaptic cell what is already there. So these can tell the presynaptic neurons if there's already enough of that neurotransmitter, we've already hit the levels that we need to hit, you can slow down release. Or maybe there isn't enough, you should increase production. So these are autoreceptors, sort of like they're regulating what's going on within that synapse. And we also have this weird case where the postsynaptic neurons can also regulate activity, which is kind of cool. But the postsynaptic neurons can change either the sensitivity of their receptors, so they can make those receptors either more or less sensitive to incoming signals, or in other strange cases, they can actually make more receptors. So if they have more receptors, then you're going to get more of a response from the same amount of signal, which is kind of cool. So you can change that receptor sensitivity or the number of receptors to compensate for either an increase or decreased amount of the neurotransmitter that's being released. If it wants to get more of a signal, then maybe it's going to be more sensitive. If it wants to get less of a signal, it gets less sensitive. And so this is what I meant by things getting more complicated as we start adding more layers to it. And so I also mentioned a little bit earlier that at this point with the neurotransmitters in that synaptic cleft that we do have types of glial cells that can surround that synapse to prevent the neurotransmitters from spilling out into the environment freely. So they can be involved in keeping those neurotransmitters where they need to be and recycling them if they need to be recycled. And in a really neat case, some glial cells can actually vary the amount of neurotransmitter that's free-floating in that cleft by changing how much they remove or don't remove. And in very special scenarios, this is, again, not super well understood, there are some glial cells that actually release their own neurotransmitters, just to make things even more complicated. But for some of these, especially the ones that I just sort of mentioned in passing, those are more of a, it's neat to note, but we don't know enough about how it works yet to spend a ton of time on it. All right, and then again, for those who are more visual people, I included the figure from the textbook that shows how we can see these different interactions of our autoreceptors and how we can change either the number or the location of receptors and things like that. So just a nice little visual to go along with what I said on the previous slide. yeah, we'll keep going here. I only have 3 more slides, so we might be a little bit ahead by the end of this. But so still talking about neurotransmitters, this is the thing that I kind of hinted that was coming. So originally, there was an erroneous belief that each neuron only released a certain neurotransmitter. So 1 neuron equals 1 neurotransmitter. And this was called Dale's principle. And we've stressed that it is an erroneous belief because we now know, again, that it's a lot more complicated than once thought. So not only can neurons release multiple different kinds of neurotransmitters, they can release them in a bunch of different ways. So we're going to look at 3 different methods. So we can look at co-release, co-transmission, and then the release of different transmitters from various terminals. And for this one, I'm actually going to go to the visual slide to talk about it because I find that a little bit more helpful here. So we have our partial vesicle opening is what co-release is. And so with co-release, you can think of it as being 2 different neurotransmitters co-housed. They are taken in the same place and they are released from the same vesicle. So we can see here we have the small red neurotransmitters and a larger blue neurotransmitter. Because our neurotransmitters are just proteins, so they can be different sizes and shapes and have different qualities. So in this case, we can have a controlled release of either the smaller neurotransmitter or both the small and the large neurotransmitter, controlled by how those vesicles connect to and join with the cell membrane. So if they join up and only create a small opening, then only the small neurotransmitters can escape. If the signal that's received says let all of them out, then it can create a larger opening and allow both out at once. But this allows us to control when and where each of those neurotransmitters is released. So we could have the smaller ones are the ones that need to be received first, and maybe the larger ones need to be received second. So you start with a smaller hole and then get to a larger one. So you can control delivery that way. Our other 2 options are actually technically a version of co-transmission. So our spatial separation that we saw on the previous slide, that release of different transmitters from different terminals, is really just a type of co-transmission. But that's how the textbook divided them, so who am I to argue? So what we can have with co-transmission is the idea of neurotransmitters being stored in separate vesicles. So here, our reds and our blues don't necessarily differ in terms of size or anything like that, but they're in different vesicles. And those vesicles are actually going to be differentially sensitive to things. So in the first example, we have a differential calcium sensitivity. So calcium's another ion that exists in the cell, and you can very finely control the concentration of calcium at those synapses. And by doing that, you can control which vesicles fuse with the cell membrane and when. So what we might have is this idea here where our red neurotransmitters are in vesicles that are more sensitive to calcium. So what that means is that at much lower concentrations of those calcium ions, that vesicle will fuse with the outer membrane and spill out those red neurotransmitters. As the calcium concentration increases, we can then have a release of the blue ones as well. So again, by controlling that one quality, that calcium concentration, we can control which of those neurotransmitters is released and when. And then our spatial segregation is a type of co-transmission because, again, the neurotransmitters are still kept in separate vesicles, but here we're saying that different axon terminals would have different neurotransmitters in them. So one terminal would have the blues and one would have the reds, so we don't have to rely on differential sensitivity or anything like that because it's a locationally controlled thing. And so all of these allow us to control what's being released, where, and when, with varying degrees of control. And this is meant to be sort of that final layer of there are lots of different things going on here, lots of different things that can work together, and it leads to a much more complex and controlled system than the basic picture that you might have heard before, which is neurons fire or don't fire. So we get a lot more into that, and these are terms and things that will keep coming up throughout the course. So for our sort of wrap up here, as we get into the end of it, I want to talk about, okay, so we have this, we can change firing and rates of firing and all of that, but what does it mean? How do we turn that into something? And if you're like me, you might be a little bit disappointed with the answer that we don't quite know. There's this idea that we have neural codes. So I mentioned the fact that neurons have sort of a background rate of firing. They're going to fire 4 times a second. And in the presence of excitatory neurotransmitters, maybe they're going to fire 10 times a second. And in inhibitory ones, it's going to be only 2 times a second, or whatever. So we can have varied intervals between those spikes that are produced by action potentials. And so we can interpret different combinations of firing and not firing to mean different things. Again, somehow. There's also the idea of neural networks, where it's not just neurons firing in a particular pattern, but it's specific neurons that are connected in specific patterns that then fire in specific patterns. So that's the idea that we have our neurons sort of connecting in a predictable way, and if you can stimulate that same kind of firing in those exact same neurons, you can create the same sensation. And there's, again, a really cool thing in the textbook where they talk about working with rats, and they looked at taste receptors. So if the rats were drinking sugar water, then a certain pattern of firing would appear in a certain part of their brain. Rats love sugar water, so that's a good thing. If they were drinking water that was sort of sour, that's less appealing, they're more likely to avoid it. And it would produce a different pattern in different neurons. And they could, by applying electrodes to those specific neurons, mimic the patterns of either sweet or sour when the rats were drinking what was actually just neutral water. And they found that by manipulating those specific pathways in the right pattern, they could get the rats to drink more from the plain water when they were thinking it was sweet, and they could get them to drink less if they were convinced that the plain water was actually sour, even if they were water deprived, which is really cool. So that's to the level to which we're talking about it in this chapter, but it's one of those things where as we're getting more and more computing power, as we're learning more about how these things connect, and as we're able to study in more and more fine detail, we're getting a better picture of what's going on. And in fact, we have a project called the Human Connectome Project, and I guess that's a play on the Human Genome Project. And they're specifically looking at trying to map all of the circuits in the brain. And this is a massive undertaking. As we've said, there are millions of connections in our brains. Actually, it's millions of neurons with trillions of connections. But it's a massive thing. So most of these successful type connectome projects are on much smaller scale. So something like nematodes, where you can map all, is it like 28 of their neurons? That's usually a good place to start. But as our capacity is increasing, we're also looking a little bit more lofty goals. So that's what they're working on now, and I don't think it's going to be finished anytime soon. All right, so our big take-home for this is it's really hard to get that big picture understanding of what's going on in the brain, and specifically the behavior that that brain then produces, without understanding what's going on at that neural level. So we have to understand the basics in order to then move on to the more complex stuff, which is why we get put through this topic early on. We also have this idea that our modern tools and this working together and combining ideas is leading us to a better understanding of what have been very not well understood topics. And so these cooperative efforts, especially looking at cooperation between large universities or large foundations, it's making it to be less work for any one individual, and that's probably going to be a continuing trend moving forward, because doing something like mapping the whole human brain is going to take a very long time. And if they can speed it up in any way, they're definitely going to do it. So hopefully we have a little bit better of a grasp of the complexity of what's going on in the brain, and we can start moving from there. Yep. I don't know off the top of my head. I would assume so, just because it is such a massive project. Whenever I've done stuff looking specifically at neurons, it's done in model species, so non-humans. Again, nematodes, lots of stuff in like sea creatures, because squid have very nicely observable neurons. But I don't know if there's any directly related to the human stuff. If I remember, I'll take a look when I go home and see, because I'm sure they'll talk about it. But yeah, I'll look into it. Cool. All right, well, I will set you guys free. It looks like we're going to be about a lecture ahead, so we'll start chapter 3 a little bit early, but I guess this means that we just have a little more time getting into the exam, the midterm, to study chapters, which isn't a bad thing.